Hello and good morning, everybody, and welcome to GemPharmatech's monthly webinar series. My name is John Jackson. I'm part of the marketing team here at GemPharmatech. And today we're going to hear from Xinjie Lian, a project manager on the Immuno Oncology team, as she discusses selecting the right humanized mouse models specific to immuno-oncology programs. Before we get started, I'm going to give you a quick introduction to GemPharmatech. You may know that GemPharmatech has the largest collection of genetically engineered mouse models, but you might not know that we are a full-service preclinical CRO that, together with the models, we also deliver data and insights that move drug discovery forward. We work with academic partners as well as biotech and big pharma, and have clients all over the world who trust us not only to provide the tools that they need to advance their research and discovery forward, but also trust us with what they're doing, as well as different types of projects like large-scale breeding projects and custom model generation. We do this through 8 different locations worldwide, and we complete over 2,000 preclinical studies each year. Today, we're going to talk about immuno-oncology, but we work across different disease areas, including neuroscience, autoimmune, cardiovascular, and rare disease. So I encourage you, if you are interested in something outside of immuno-oncology, to follow us on LinkedIn, to stay up to date on our webinar series where we continually discuss different topics. And if you have a topic that you want to hear about in the future, I encourage you to, let us know either on LinkedIn or you can pop a message in here later in the webinar. The last thing before we get started is I'm excited to invite everybody, but most likely this is most relevant to people in San Diego. If you are joining us from San Diego, we are having our annual symposium in September, hosted at the Alexandria at Torrey Pines. And this year's focus is on metabolic disease. And you can hear from scientists and researchers both at academia and industry, including the Touchstone Diabetes Center and Eli Lilly. So if you are here and you want to come out and listen to some of the leading voices in metabolic disease research and development, I encourage you to register today. Space is limited and it's a completely free event. So shifting back to immuno-oncology, I will let Xinjie take it from here as she discusses today's topic of selecting the right humanized models for your immuno-oncology program.
Okay, thank you, John. In the next 40 minutes, I have broken our presentation down into 3 parts. Part 1, the importance of model selection. Part 2, advantages of humanized models. Part 3, comparison of different humanized models. Let's start with a fundamental question. Why do we actually need humanized mouse models? To answer this, let's reference a landmark benchmark study published in Nature Immunology. The authors set out to answer a question that plagues every biotech founder and head of pharmacology. To what extent do standard mouse models actually reflect the tumor immune microenvironment of real cancer patients? They systematically analyzed 15 of the most widely used syngeneic murine tumor models and mapped their immune cell composition directly against the clinical patient biopsy database. The finding was striking. In human solid tumors, T cells are the primary driver of anti-tumor immunity and the primary target of the most checkpoint inhibitors. In standard murine tumor models, the TME is heavily biased toward myeloid cells, specifically macrophage enrichment. In fact, over 16% of conventional mouse tumor models biologically align with what clinicians classify as immunodesert or macrophage enrichment subtypes. Think about what that means for your candidate molecule. If you evaluate a novel T-cell engager or anti-PD-1 antibody in a standard mouse, you are testing a T-cell drug in a myeloid dominated microenvironment the host and the drug are speaking entirely different biological languages. This fundamental mismatch is one of the single biggest reasons why so many immune oncology therapeutics look like miracle cures in preclinical studies, only to flop in phase 1 and 2 human trials. Humanized models were created especially to bridge this translational divide. If the theoretical argument isn't compelling enough, let's look at a concrete real-world case study. On this slide, you see 4 sides of tumor growth curves. Every single curve evaluates the exact same clinical stage anti-PD-L1 antibody in phasing. The drug, the dosing frequency, and the concentration remained completely identical across all arms. The only variable altered was the humanized model background. Group 1. In phasing tested in a Y-type murine background, the treatment curve completely overlaps with the vehicle control. The result was zero anti-tumor efficacy. Group 2. Y-type mouse implanted with PD-L1 humanized tumor cells. The result showed moderate partial efficacy. Group 3. PD-L1 humanized host mouse implanted with Y-type mouse tumor cells. The result was again partial efficacy. Group 4. PD-L1 humanized host mouse implanted with PD-L1 humanized tumor cells. The result showed statistically significant tumor regression. If a drug developer had evaluated inflation solely on the first model background, they would have concluded the molecules was inactive and killed the program in early discovery. In preclinical immuno-oncology, humanized models are not passive tools. They are active biological variables that directly dictate your data outcomes. Knowing what a model can and cannot answer is far more critical than simply generating a pretty tumor suppression curve. Now that we have established the necessity of these systems, let's explore part 2, the mainstream humanized model platforms available today. All humanized mouse models fall into 2 master categories based on their host immune background. Category A, immune deficient reconstitution models. Here, we start with a severely immunodeficient mouse, typically on our severe immunodeficient NCG background. We then engraft human peripheral immune cells or hematopoietic stem cells to build a human immune system inside the mouse. This category includes human PBMC NCG, human HS NCG, cytokine knock-in strains, and HLA humanized models. Category B, immunocompetent target humanized models. Here, we take a fully functional immunocompetent mouse string, like B6 or BABC, and use precise gene editing to replace a specific mouse protein with its human counterpart. The rule of thumb for choosing between these 2 categories is straightforward. If your scientific hypothesis hinges on how human T cells, NK cells, or macrophage interact with tumors in vivo, you need an immunodeficient reconstitution models. If your goal is to test a target-specific binder, or bispecific antibody in the presence of an intact, fully functioning immune system without GVHD, you need a gene-humanized immunocompetent model. Let's examine each of these 5 platforms individually. Platform 1 is the human PBMC-NCG model. This is the workhorse of rapid immune checkpoint and T-cell engager screening. Construction is remarkably simple. We isolate adult human PBMC and inject them directly into NCG mice via intravenously or intraperitoneal routes. No preconditioning irradiation is required. Once injected, mature human T cells undergo robust expansion driven by foreign mouse tissue cues. By week 2 to 3, human CD45 positive cells rise steadily, reaching an operational plateau around week 4. Over 90% of these reconstituted cells are functional human CD3 positive T cells. The core advantages are speed and cost efficiency. You achieve full T cell reconstitution in just 7 to 14 days. It requires minimal pre-incubation and now specialized housing or radiation equipment. The biological bottleneck is GVHD because we are transferring mature immunocompetent human T cells into a mouse host. These T cells eventually recognize host murine tissues as foreign, triggering GVHD. GVHD symptoms typically emerge around week 5 post-transplant. Therefore, the experimental window for the human PBMC-NCG is 2 to 4 weeks. It's ideal for short-term assays, screening immune checkpoint inhibitors, bispecific T-cell engagers, and short-duration vaccine regimens. Okay, let's look at a real-world validation data generated on the human PBMC-NCG platform. Left panel, we tested the anti-PD-L1 antibody Tecentriq across 2 independent human PBMC donor cohorts. In donor 1, Tecentriq achieved a tumor growth inhibition of 45%. In donor 2, TGI reached 56%. Notice how donor-to-donor variability reflects real human heterogeneity, while maintaining robust anti-tumor signals. Red panel, we compared single agents against a bispecific molecule. PD-1 monotherapy showed minimal efficacy. Avastin monotherapy showed moderate response, but the AK112 PD-1 VEGF bispecific antibody demonstrated a clear superiority with a TGI of 75%. Notice that both study timelines were completed within 20 to 30 days. As long as your study protocol is designed within this effective window, human PBMC-NCG provides actionable data. Beyond the traditional systematical monoclonal antibodies, human PBMC-NCG has become a gold standard platform for in vivo CAR modalities. This slide features data from a landmark science paper. Instead of extracting T cells, engineering them ex vivo, and re-infusing them, Next-generation platforms use lipid nanoparticles carrying mRNA to directly engineer T cells inside the patient's body. The experimental design was day -18 human PBMC transplanted into NCG mice. Day -8 NAM6 leukemia cells inoculated. Day 0 LNP mRNA therapeutics administered. Flow cytometry data shows that LNP successfully transfects circulating human T cell in vivo, producing peak CD8 positive CAR expression at day 7. Bioluminescence image proves that this in situ CAR generation leads to clear, dose-dependent clearance of the NAM6 tumor burden. Because mature T cells in the human PBMC NCG are circulating in a relatively quiescent state, it serves as an ideal testbed for confirming LNP tissue targeting, viral transduction efficacy, and short-term toxicity, cytotoxicity. However, many cell therapies, therapeutic vaccines, and solid tumor regimens require longer than 3 weeks to demonstrate a complete mechanism of action. How do we eliminate the GVHD barrier while retaining the quick reconstitution of PBMC transplantation? The answer lies in our double knockout platform, human PBMC NCG MHC double KO. By knocking out both MHC class 1 and MHC class 2 molecules on the NCG background, host tissues no longer present major histocompatibility antigens to incoming human T cells. Look at the timeline on this slide. Human PBMCs were engrafted on day -1, tumor cells inoculated, and the mice were monitored out to day 80, nearly 3 months, with 0 signs of lethal GVHD. The vehicle control group maintains stable, robust tumor growth over time. If your program requires testing in vivo CAR, long-acting bispecific or multi-dose combinations over 6 to 10 weeks without xenogenic inflammatory background noise, the MHC double KO variant is the optimal choice. Okay, let's now move to platform 2, human HSC NCG. While PBMC models prioritize speed, HSC humanization prioritize physiological completeness and long-term durability. Rather than using mature peripheral blood cells, we isolate human CD35 positive hematopoietic stem cells derived from umbilical cord blood. Recipient newborn or young NCG mice receive sublethal irradiation to clear native bone marrow niches. CD34 positive cells are then infused intravenously. Over a 10 to 12-week incubation period, these stem cells differentiate inside the mouse bone marrow, producing a de novo human immune system. Functional human CD3 positive T cells educated in the host thymus developed human CD19, CD20 positive B cells. monocytes and naive lymphocytes populations. Here is the key metrics of human HSNCG. First, minimal GVHD, enabling operational experimental windows of 18 to 26 weeks. Second, to bypass the 12-week setup delay for our clients, GemPharmatech maintains large rolling monthly cohorts of pre-reconstituted, quality-controlled human HSNCG mice ready for immediate study enrollment. Let's examine how human HSA-NCG performers across complex oncology pipelines. Left panel, evaluating a novel tri-specific T-cell engager against Blincyto. Both tri-specific T-cell engager and Blincyto demonstrated highly significant sustained tumor inhibition across a 30-day treatment window. Right panel, testing dose-dependent responses of Avastin. The 1.6 milligrams per kilogram group yield 16.2% TGI, while the 3.25 milligrams per kilogram group achieved 67% TGI, demonstrating linear pharmacology. Furthermore, because human HSA-NCG reconstitutes human B cells alongside T cells, it provides an invaluable model for evaluating in vivo CAR-T targeted against hematological markers like CD19. As shown in the lentiviral and LNP vectors on the screen, researchers can simultaneously monitor target tumor detraction and normal B cell aplasia within the extract same animal. Okay, where standard human HSNCG models successfully rebuild T cells and B cells compartments, standard immunodeficient mice lack key across species cytokines required to support human NK cells and myeloid lineages. This brings us to platform 3, human cytokine knock-in models. To solve lineage-specific dropouts, GemPharmatech engineering the target human cytokine genes directly into the NCG genome. As shown in our platform map, we have developed a dedicated lineage suite. NCG human IL-15 expresses human IL-15 to driven NK cells lineage commitment. NCGM expresses human SCF, GM-CSF, and IL-3 to driving myeloid differentiation. Macrophages, monocytes, disease. Let's look closely at how NCG human IL-15 and NCGM transform preclinical evaluation. NK cell therapeutics, NK cell engagers, and ADCC-enhanced antibodies require functional, long-lived human NK cells. Standard NCG mice cannot provide this because murine IL-15 does not cross-react effectively with human IL-15 receptors. In the NCG human IL-15 strain, human IL-15 is continuously expressed, providing the necessary survival signals. This strain offers three versatile reconstitution strategies. First, purified human NK cell transfer, ultra-fast setup, 3 to 5 days to study start. Ideal for rapid screening of NK cell engages. PBMC transfer into human NCG human IL-15 yields coexisting functional T cells and NK cells within 7 to 14 days. Great for comparing T versus NK dual bispecific. Third, CD34 positive HLC transfer into NCG human IL-15. The gold standard produce robust multilineage T, B, and fully mature CD 56 positive NK cells that persist for over 18 weeks. Flow cytometry confirms high frequencies of functional human CD56 positive NK cells in peripheral blood without causing systemic toxicity or weight loss in the host animal. Okay, here is the proof of performance for human HSC-NCG, human IL-15. Left panel evaluating an antibody targeting NKG2A, a key inhibitory checkpoint expressed on NK cells and CD8 positive cytotoxic T cells. Monotherapy treatment demonstrated a clear anti-tumor efficacy, achieving a TGI of 50%. In standard NCG mice lacking NK cell support, this drug shows minimal activity. Right panel, side-by-side assessment of an ADCC mediating anti-CD20 antibody, rituximab, versus a T-cell engager, Blincyto. Rituximab achieved 59% TGI via NK-mediated ADCC, where Blincyto achieved 48% TGI via T-cell redirection. This single model allows you to benchmark T-cell modalities directly against the NK cell modalities in an environment where both effector cell types are active. Now let's turn to NCGM, our human myeloid supportive strain. Myeloid cells including tumor-associated macrophage, DCs, and myeloid-derived suppressor cells constitute a massive portion of the human tumor microenvironment. However, in standard HLC reconstituted mice, human CD33 positive myeloid cells make up less than 1 to 2% of human leukocytes. By knocking in human SCF, GM-CSF, and IL-3, the human HSC-NCGM model dramatically expands human CD33 positive myeloid lineages. Look at the tissue distribution bar graphs on this slide, where T cell and B cell levels remain consistent with standard NCG. Human CD33 positive myeloid cells increase significantly across peripheral blood, spleen, and bone marrow. This makes human HSC-NCGM the premier model for drugs targeting myeloid immune checkpoints, such as CD47, SPA alpha, ILT2, ILT4. Next, on this slide, we evaluate a candidate drug targeting the ILT2 and ILT4 immunosuppressive myeloid pathway in SK-MEL5 melanoma-bearing human HSC-NCGM mice. After enrolling mice with greater than 25% human CD45 positive engraftment, dosing was initiated. The test drug achieved a tumor growth inhibition of 24% relatively to the isotype control. Now, I want to show an important nuance regarding data interpretation. In T-cell-rich models, checkpoint drugs often yield dramatic greater than 60% GGI values. However, myeloid checkpoint inhibitors do not directly induce hypercytotoxic T-cell bursts. Instead, they relieve local macrophage suppression and restore phagocytosis. A 24% single-agent TGI in a myeloid model is a biological meaningful signal, indicating functional targeting engagement in vivo. In clinical practice, these targets are designed to be paired with cytotoxic antigens or T-cell checkpoint. Let's move to platform 4, HLA humanized models. This platform addresses a critical immunological constraint, MHC restriction. In standard human HSC-NCG mice, human T-cell progenitors are educated in the host mouse thymus. As a result, their T-cell receptors are educated on murine MHC molecules. If your therapeutic pipeline involves TCR-T therapies, new antigen cancer vaccines, or HLA-restricted bispecifics, standard humanized mice will fail. Because these drugs require human T cells to recognize specific peptide fragments presented strictly on human HLA complexes. We knocked the human HLA A2.1 gene into the mouse MHC class 1 locus. When CD34 positive stem cells form an HLA-A2 matched human donor are engrafted into NCGM human HLA-A2.1 mass. Human CD8 positive T cells mature in an HLA-A2 restricted manner. These T cells cannot properly recognize tumor antigen peptides represented by human HLA-A2 molecules on PDX. This slide demonstrates this in an HLA-A2.1 matched PDX tumor study. Irradiated NCGM human HLA-A2.1 mice were transplanted with HLA-A2 positive CD34 positive HSC. An HLA-A2.1 positive human PDX was implanted. Animals were treated with normal therapies versus a novel combination regimen. The data shows that the combination therapy group 4 achieved potent, sustained tumor suppression compared to monotherapies. Because both the host immune system and the tumor tissue shared matched HLA-A2 restriction, The observed T-cell cytotoxicity represents genuine antigen-specific killing. For TCR-T and new antigen vaccine developers, this model provides indispensable IND-enabling proof of concept. Finally, let's discuss Platform 5, target gene humanized models. Unlike the four purines platform, these mice are fully immunocompetent. We do not perform cell transplantation, and there's risk of GvHD. Instead, we use CRISPR-Cas9 engineering to replace specific murine protein domains with human protein coding sequences in situ. GemPharmatech has engineered hundreds of single, double, triple, and quadruple gene humanized strains. Single targets includes human PD-1, human TIGIT, human LAG-3. Double targets includes human PD-1, CTLA-4, human CD47 SPA alpha. Quadruple knock-ins such as human PD-1, PD-L1, CD47 SPA alpha. For multi-specific antibodies, this platform has three core advantages. First, preserve the physiology. Complete murine immune network intact. Lymph nodes, spleen architecture, natural cytokine cascades. Second, safety and toxicity assessment. Superior model for evaluating immune-related adverse events and cytokine release syndrome. Third, surgenic cell line libraries paired with matched target humanized most tumor cell lines. For example, MC38 human PD-L1, CD26 human CD47. Okay, let's now turn to the efficacy data for immune checkpoint inhibitors on our target humanized mouse models. For this study, we used B6 PD-1 PD-L1 dual humanized mice implanted with MC38 tumor cells that we engineered to overexpress human PD-L1 at the same time knock out mouse PD-L1. Why did it do that? Simple. We wanted the PD-L1 on the tumor cells to be entirely human, so it matches Keytruda's target perfectly. And we also wanted to rule out any cross-reactivity with mouse PD-L1. If you look at these two graphs, you will see very nice dose-response curves. Keytruda gives a clear dose-dependent efficacy gradient, and the Tecentriq shows the same, a nice, clear dose response. These two datasets tell us that the B6 dual humanized model is a sensitive, stable, and highly responsive platform for evaluating PD-1, PD-L1 pathway drugs. Now, let's move to efficacy data for an IL-12-related drugs. Using our RBC human IL-12-RB1, IL-12-RB2 dual gene humanized model, you might ask, why do we need to humanize both IL-12 receptor subunits? Because IL-12's biological function requires both IL-12-RB1 and IL-12-RB2 to form a function receptor complex. If you only humanize one and leave the other murine, human IL-12 drugs simply can't bind and activate the signaling pathway effectively. So dual humanization here is the prerequisite for any meaningful evaluation of IL-12 pathway drugs. In this experiment, we used the BABC background dual humanized mice implanted with the CT26 colorectal cancer sugenic model. The slide shows two independent efficacy studies. These two datasets demonstrate that this BABC dual humanized model can effectively support efficacy evaluation of IL-12 pathway drugs. And moreover, it can clearly distinguish potency difference between different molecules. One more point worth highlighting. IL-12 class drugs activate multiple immune cell types and trigger cascades of cytokine release. Evaluating them in immunocompetent gene-humanized models gives you a much more realistic picture of systemic and microenvironment modulation than using immunodeficient reconstitution models. And on top of that, these models also let you pick up early toxicity signals, which is very helpful for planning downstream safety studies. Early, we introduced the HLA humanized models on the NCG background. Now let's look at HLA humanized models on an immunocompetent mouse background. This slide shows an immunogenicity evaluation of an mRNA vaccine in an HLA humanized model. We used the CB6F1, that's an F1 hybrid of B6 and BABC, and introduced the human HLA A2.1 into that background. Here is the timeline. Day 0. Intramuscular injection of the mRNA vaccine. Day 14. Second injection. Day 28. Third injection. Day 42. Endpoint collection for elastiport acids to measure cellular immune responses. We had 3 groups. Group 1, vaccine candidate 1. Group 2, vaccine candidate 2. Group 3, saline as a negative control. For the readout, we used multiple peptide pools to stimulate splenocytes and then elastiport to direct IFN gamma secreting T cells, maturing support-forming units to quantitatively assess the vaccine-induced antigen-specific T cell response. Now, look at the bar graph on the right. The saline control has very long background, exactly what you'd expect. Both vaccine groups, Group 1 and Group 2, show SFU significantly above control, meaning both mRNA vaccine successfully induce antigen-specific T cell responses. And interestingly, there's also a clear difference between Group 1 and Group 2, so this gives you direct head-to-head evidence for comparing immunogenicity of different vaccine candidates. This is a classic application that really highlights the core value of HLA humanized model in vaccine development, because the antigen encoded by the mRNA vaccine have to be presented by HLA molecules to be recognized and activate T cells. Without an HLA humanized model, you simply can't recapitulate that HLA restricted antigen presentation process in vivo. With it, you get data that actually translates to clinical. Now, let's put our 5 platform categories together into a consolidated decision framework. When planning your preclinical immune oncology strategy, refer to this side-by-side comparison matrix. When mapping your pipeline to this matrix, ask yourself 3 simple direction questions. 1. Do I need a fully intact immune system to assess systemic safety or murine-matched tumor kinetics? Choose target gene-humanized models. 2. Does my lead candidate target human NK cell, macrophages, or HLA-restricted TCRs? Choose cytokine or HLA-humanized platforms. 3. What is my project deadline? If you need drug ranking data next month, select human PBMC NCG. If you are building an IND dossier over the next 6 months, select human HSC NCG. To summarize our session today, model selection dictates data outcomes. As we saw with infusing, picking the wrong model and falsely kill a variable drug or validate an inactive compound. Match the model to your drug's mechanism of action. There is no single perfect humanized mouse. PBMC models provide rapid T-cell insights. HLC models provide multilineage dips. Cytokine knock-ins restore NK and myeloid cells. HLA strengths unlock TCR-T therapies, and gene-humanized mice provide immunocompetent safety profiles. Plan ahead for IND studies. Align your timelines early with model reconstitution requirements, so your preclinical package is robust, reproducible, and ready for regulatory scrutiny. At GemPharmatech, our goal is to partner with you to select the customize these traditional tools to accelerate your pipeline into the clinical. Thank you very much for your time.
Thank you, everybody, for joining us. Thank you, Xinjie, for speaking. As I said, follow us on LinkedIn to get more updates on future webinars. Sometimes we have very specific topics. Sometimes we have kind of overarching topics like this. So definitely give us a follow, and thank you for joining us. Also, again, if you're in San Diego, consider registering for our symposium in September. With that, we'll sign off. I hope everyone has a great day, and we will see you next month. Thank you.